Hey, in this video, we're going to talk about phospholipids. Okay, so we should be familiar with these lipids, triglycerides, which are the ones that most of the lipids, the fat that we take in our diet is in the form of triglycerides. That you store the fat in our bodies as triglycerides, and that's made up of the glycerol molecule. And we have ester linkages to 3 fatty acids. Okay. We'll have a look at the structure in that more detail. Now, phospholipid is very similar. So here we've got the phospholipid. You'll see that we've only got 2 fatty acids there, okay, which leaves one of the oxygens of the glycerol molecule is free, and that joins onto a phosphate group, which is charged, and a choline group, and that contains a nitrogen atom. That's what we'll put an N there. So we'll have a look at those. Have a look at the atoms in that in more detail now. But if you just remember this structure, really, this is all you really need to know. But just to put a little bit of detail into that so we know what's going on. Okay, so this yellow bit here, right, so we've got our glycerol, yeah, glycerol attached there. We've got the yellow bit and the red bit, right? The yellow bit is called choline. And you can see that it contains a positively charged nitrogen atom. If you look at that nitrogen is forming 4 bonds. Nitrogen only usually forms 3 bonds, and it's whenever it does form 4 bonds, it's always got a positive charge in it, nearly always. You see it's got a positive charge on the nitrogen, and that's important because it makes it very hydrophilic, makes it soluble in water. When things are charged, they tend to be soluble in water because water is called a polar solvent. And then attached at the other end of the choline molecule, we've got this phosphate group here in red. Okay, now the phosphorus atom there you can see is forming 5 bonds. Phosphorus always forms 5 bonds, nearly always, sometimes 3, but usually 5 bonds. And again, we've got a negative charge on the oxygen in the phosphate group, which is going to make that pretty water soluble. It's going to make it hydrophilic, water loving. And then we've got the glycerol molecule. Right, just to show you, here's a slightly more convenient way of doing it. Instead of having this CH3 here, instead of drawing all the bonds, usually you just write that down. As you can see, it's CH3, which makes it a bit easier to see. Also, I've just so I put some I'll just shrink this a little bit so you can see what's going on. Okay, right, you can see there. Okay, we've got our this is our the whole phospholipid molecule. So in yellow up here we have the choline. Yeah, there's the choline, positively charged, dissolving in water. In red we've got the phosphate, negatively charged. That means that's going to be soluble in water. In green we've got the glycerol molecule, and you have 2 ester linkages to these blue things, which are the fatty acids. So the ester linkage is that bit there. Right, that's the ester, the C double bond O and the O. And then the blue zigzag bit, and if you remember, that it's to save me drawing it out, that is that's basically you've got lots of carbons with 2 hydrogens attached, and they are those fatty acids. They're long. That's typically about 18 carbons long, between 14 and 20, but usually about 18 carbons long, pretty long. Now, that's important because this part of the molecule is going to be, it's all carbons and hydrogens. That means that is going to be hydrophobic. That will not be water soluble at all. So we have got a molecule, it's sometimes called amphipathic, and it means it's got a hydrophilic head up here and a hydrophobic tail, the double tail really, isn't it? Because you've got 2 fatty acids, 2 of those blue fatty acids stuck onto the glycerol. So that's a phospholipid molecule, and they're really very important molecules because they make up cell membranes. Okay, anyway, we'll talk about that in 1 minute. Well, you can see the picture of the cell membrane coming up here. Well, let's have a look. So what we got there, that red bit, that is the hydrophilic head. So that is this bit here, the choline and the phosphate is charged, okay? And the blue, the double blue thing is obviously the fatty acids, okay? So that's hydrophobic. Right, now this is the cell membrane. So what we've got out here, this is inside the cell, and this is outside the cell. Now, inside the cell, you're going to have it's called an aqueous environment. Basically, everything's dissolved in water. Outside the cell is also an aqueous environment. Everything's dissolved in water. So you can see the hydrophilic heads. What they do is they try and get close to the water, so they'll stick outwards. Whereas these hydrophobic tails, they hate the water. They want to get away from the water. They like each other. They form strong interactions with each other. So they point inwards, and this is make this a little bit bigger. Okay, so this is called the Phospholipid Bilayer. It's called a bilayer because obviously there's two layers of phospholipid molecules, and it's all due to the fact that they've got these hydrophilic heads and hydrophobic tails. In fact, if you get some pure phospholipid in a test tube, shake it up with water, it will naturally form into these bilayers. It will naturally do that. Okay, and that's so you got your cell. Here's my cell with a nucleus, you know, and there's the cytoplasm, which is aqueous, and the black line is the cell membrane, and that is made up of a phospholipid bilayer. Really important. It's got the hydrophilic heads and hydrophobic tails. Also, what we've got in there is we've got this green thing, okay, and this is a protein. Okay, so you do have proteins, integral proteins, embedded in the phospholipid membrane, and they do all different sorts of jobs. So, for example, it may be a channel. So it could be like have a path where an ion can pass through from the outside into the inside. It could be a site that recognizes a hormone, so a hormone may bind to this end here, and then that causes changes down at this end of the molecule, and then transmits information to the inside of the cell, telling the cell that, you know, insulin or whatever has bound to the outside, and that causes changes within inside the cell. So membrane proteins do lots of different functions, lots of really important functions, which you'll find out about later. Okay, just one last thing is we can see some of these phospholipid molecules have a fatty acid with a kink in it, like that one there. Okay, now what is that caused by? That is caused by, if we look here, we have got a double bond, right? So that means it is unsaturated. And whenever you have a fatty acid with a double bond in it, it produces this kink. So there's a double bond. Most of the carbons have two hydrogens attached in the fatty acid. So these ones have two hydrogens. Of course, carbon only forms 4 bonds, so these two here will just have 1 hydrogen. That's called an unsaturated fatty acid, and as I say, it produces a kink. Now that's pretty important because you can see where you do have a kink, such as there, they don't actually pack together very well. These the hydrophobic tails of the phospholipid molecules don't pack together, and that's pretty important, because if they do, the closer they pack together, the stronger the interactions between one phospholipid molecule and another. And if the interactions are too strong, the membrane is not fluid enough. Okay, it's not fluid. It means it's too rigid. It needs to be fluid, and so you need unsaturated fats, unsaturated fatty acids, in order to keep the membrane sufficiently fluid. Also, to help with fluidity, you have another molecule, which I'll draw in green here, which sort of fits in. It's a hydrophobic molecule, so it's going to fit in there. So that yellow one there. You can tell it's hydrophobic because that's going in the hydrophobic part of the membrane, and that, you will have heard of the name of it, even if you don't know what it is, cholesterol. So cholesterol is also important in certainly in animal cell membranes, in keeping them fluid. So you have unsaturated fatty acids and you have cholesterol. They're quite important in keeping the membrane fluid. And don't forget, it's called a phospholipid bilayer. And that's why phospholipids are so important. Every cell, of course, is covered by a membrane, which is made within a phospholipid bilayer. And you'll also know that there are membranes inside cells. The mitochondria has a membrane around it. Vesicles have membrane. The Golgi apparatus is made of membrane. The rough endoplasmic reticulum. So membrane is a very important part of all cells.